Hello and welcome to the status of recombinant reagents used for endotoxin testing, presented by American Pharmaceutical Review and sponsored by Associates of Cape Cod. My name is Brendan Raher, Director of Creative Services at American Pharmaceutical Review, and I will be the moderator during today's event. The agenda for today's webinar includes this introduction, followed by the presentation, and we will conclude this event with a live question and answer session. To ask a question, click on the Ask a Questions tab on the right side of your screen. Please take a note, the right side of your screen also features an overview of this webinar and more information regarding your speakers. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use social media widgets beneath the webinar to share with your friends and colleagues. And now, let me introduce today's speakers. Veronica Willis has been working in the endotoxin industry for the past 12 years and currently manages the Technical Services Department at Associates of Cape Cod. Veronica is a specialist on testing complex samples, method development, regulatory aspects of endotoxin testing development, and delivery of educational and technical contents. She attends and speaks frequently at scientific conferences and seminars globally and is an established expert on endotoxin testing. Veronica holds a master's degree in biochemical engineering from the Institute of Chemical Technology in Prague, Czech Republic. Karen Zink-McCullough is owner and principal consultant at MMI Associates, a consulting firm founded in 1985 specializing in pharmaceutical microbiology, endotoxin testing, and quality system analysis. Karen has 45 years of experience in the pharmaceutical, medical device, biologics, and cell gene therapy industries, having held leadership positions as Director of Microbiology at Organon, Senior Director of Quality Compliance at Roche Molecular Systems, and Vice President of Quality Operations at Dandrion. Karen received her BA in Bacteriology from Rutgers University and MS in Molecular Biology from the University of Oregon. Go Ducks! She is a co-founder of the LAL Users Group and is a member of the USP Expert Committee, Microbiology General Chapters. Karen is the editor of The Bacterial Endotoxins Test, a Practical Guide, and has authored 21 books and chapters and over 20 peer-reviewed and trade journal articles. She is a frequent speaker at national and international symposia and training courses on endotoxin testing, pharmaceutical microbiology, and quality systems. Veronica and Karen, please take it away.
All right, well, thank you, Brendan, and go Ducks. And I want to thank Brendan and APR and Associates of Cape Cod for asking me to participate in this webinar. What I'm going to talk about is the current USP perspectives on recombinant reagents for endotoxin testing. And before we get into this a little bit, I think that we need to understand a little bit about how USP works. So you know that USP and FDA are two different organizations. USP holds the standards and writes the test methods, and FDA is the regulatory agency. So when we talk here about these USP chapters, we're talking about chapters that are enforceable, okay, meaning that FDA can enforce the chapters. So just keep that in mind as we go along. I want you to understand that even though I put the presentation together, I didn't run it by Dr. Radhakrishna Tirumalai, Radha as we call him. He is our liaison from USP, and I ran all of this by him to make sure that, in fact, everything that I wrote here was accurate. So, again, this presentation has been reviewed by people at USP. So the bacterial endotoxins test, which is really USP 85, describes the testing and using reagents that are manufactured currently from the circulating blood cells of the horseshoe crab. In the United States here, it's Limulus polyphemus. Over in Asia, okay, there are a couple of different genera of horseshoe crabs that are being used for this purpose. The endotoxins test really describes three different kinds of assays. There's the gel clot limits test, which basically says, okay, do I meet the specification or do I don't? Do I meet the limit that the test sets or do I not? There's the gel clots assay test, where you make a series of dilutions of your product and you look for the end point. the end point being the last positive tube that's followed by a negative tube. And then you figure out how much endotoxin is your sample from the end point. There is an endpoint chromogenic test. The endpoint chromogenic test was the first of the quantitative tests that was introduced years ago. And what happens here is you measure a color reaction as a function of the endotoxin concentration. So the intensity of the color is a function of the endotoxin concentration. And what you end up with is a graph with a positive slope, because the more endotoxins activity you have, the more color you have. And so you end up with something with a positive slope. This test actually is begun and then it is stopped chemically. So the different standards and the different samples, the color reaction will be stopped at the point where it's at. The standard curve is constructed and the test results are interpolated from the standard curve. Then there's the kinetic testing, both chromogenic and turbidimetric, in which we say, okay, let's set an OD, an optical density. In the case of a chromogenic test, it's a color intensity. In the case of a turbidimetric test, it's an optical density. And let's ask how long it takes for each of the samples and standards to reach this magic optical density or magic color intensity. And then what we do is we plot the log of the number of seconds it takes for these samples to reach those optical density for the standards and samples as a function of the log of the endotoxin concentration. And what we end up in this case is a straight line with a negative slope. So the lower endotoxin concentrations take the longest to reach the magic OD, and so we end up with a negative slope. Now, historically, the reagents used in the bacterial endotoxins test have been approved by the United States government. These lysate manufacturers have BLAs, biological license applications, and they are inspected by CBER, the Center for Biologics Evaluation and Research. Now, you might say, well, why is that? Well, back in the day, they were considered to be a blood product. Not a human blood product, a horseshoe crab blood product. And so therefore it is regulated as such. But now things have evolved. And so now we've come into a recombinant reagents. And from a USP standpoint, what do we mean? So we mean that recombinant means that the formulation components of the reagent are created using recombinant or cloning technology. Now, you got to understand that when the other reagents are made, when the current reagents are made, they take the amoebocytes, the lysate manufacturers take the amoebocytes from the horseshoe crab, and they burst them open, hence the word lysate, limulus amoebocyte lysate. So what you have in the formulation of the product is not only the proteins that are important for the product for the reaction to happen, but you also have a bunch of other stuff that is in the cells, in the amoebocyte cells. Now with these recombinant reagents, we don't have all of that because we're not making these reagents from live horseshoe crab cells. We are actually cloning the critical components of the reagents. And so now the reagents in that sense are much more pure, or certainly much better qualified. There's the RFC recombinant factor C, meaning that the formulation contains only the recombinant factor C protein. And we'll see in a minute where recombinant factor C fits into all of this. And then there's the recombinant LAL reagents. And Veronica will speak to that. There's a couple of manufacturers now that have recombinant reagents, meaning that all of the proteins in this sequence have been cloned and included in the formulation. And so the question is now, are they the same as the test that's currently in the USP? From the actual running of the test, the answer is yes. There's a standard curve. We have to interpolate from the standard curve. And so the actual test methodology is the same. The only thing that's different is the reagents. So if we take a look at the classical cascade, you can see that endotoxin comes into the system there at the top. And what it does is it takes factor C, which is the protein that from recombinant factor C is made from. Factor C, and the endotoxin activates factor C. That's what that little line over the C means. The activated factor C activates factor B. Factor B activates the pro-clotting enzyme to make the clotting enzyme, and then the clotting enzyme takes the coagulagen, this is the clotting protein, and what it does is in the case of a turbidimetric, it cleaves the coagulagen to make coagulin. The coagulins stack up sort of like Tinker Toys or Lego blocks, and they stack up and accumulate, and there is an increase in turbidity here. And that's where the kinetic turbidimetric comes from. In the gel clot test, if you have enough coagulin stacked up to make a gel, all right, that's where the gel clot test comes from. In chromogenic, it's a little bit different. The factor C is the same, factor B is the same, the pro-clotting enzyme is the same. But what happens here is that rather than to have a coagulagen molecule, okay, we have a chromogenic substrate, okay, which is a substrate to which a chromophore has been attached, and the same serine protease, that is the clotting enzyme, then releases the chromophore, and the chromophore then turns yellow. So the turbidimetric and the chromogenic test are really the same thing. Okay, the reaction is exactly the same. The only thing that's different is we're measuring a different signal because the signal comes from a different source. In recombinant factor C, now look at this. These were taken from, there was a USP, a Pharmacopeial Forum article written by people at Lonza a number of years ago, and these charts were taken from that. But if you look at the recombinant C, what we have is recombinant factor C, and that gets activated by endotoxins, and then all of a sudden we're down to the substrate step, and then a fluorogenic substrate is cleaved, and then we have the product fluoresces, the reagent fluoresces at that point. Now, if you look back here to the previous slide, we have factor C, we have factor B, we have the pro-clotting enzyme and the clotting enzyme. It's a cascade. And what happens every time one of these reactions happens in the cascade, the signal is amplified a bit. In the recombinant factor C, that cascade is gone. Okay, and we just rely on the fact that this activated recombinant C is now going to go to the fluorogenic substrate, and it's going to produce fluorescence. Again, the fluorescence at this point is going to be directly related to the endotoxin's activity going in. So in all of these cases, the level of signal that comes out in the end is directly related to the endotoxin's activity that starts the reaction. In the recombinant LAL cascade, you can see that we have recombinant factor C, recombinant factor B, a recombinant pro-clotting enzyme, and then down here, it's a chromogenic test. Essentially, it's a chromogenic test. We have a chromogenic substrate, and the clotting enzyme cleaves off the chromophore, and then you get color in the end. So the recombinant LAL cascade has all of the important proteins in it from the entire cascade, and it's a chromogenic test. So this test actually ends up looking exactly like a kinetic chromogenic test. This test ends up looking like an endpoint test, right? It has a positive slope, and there's a direct relationship between the endotoxin and the fluorescence. Down here, we've got all of the cascade, and so what we have is a standard chromogenic test, run the same way. Why even start to think about recombinant reagents? Well, there's a sourcing issue, and there's a lot of discussion out there right now about the source. There's significant horseshoe crab depletion in Asia, but it really isn't so much in the United States. There's been a number of studies that have been published to show that, in fact, the horseshoe crab population in the United States is doing quite well. So there really isn't an issue with horseshoe crab depletion in the United States. But the question here is, if the horseshoe crabs are depleted in Asia, will there be an increased demand on the horseshoe crabs here in the United States, which then might have some impact on their population? So, there is a little bit concern about that. recombinant reagents aren't sourced directly from animals, and so therefore they wouldn't bother the horseshoe crab population. Recombinant reagents, in theory, could be a lot more consistent. The reagents don't rely on the variability of the individual horseshoe crabs that make up a batch of lysate. It takes quite a number of horseshoe crabs, the blood from a number of them, in order to make a batch, and it becomes seasonal. This is done in the summertime, so the whole thing is seasonal, and the whole thing depends on, to an extent, on the population that's used in order to make the batch, and so there is considerable batch-to-batch variability. In the case of the recombinant reagents, all right, the idea here is to say, okay, if we can clone these proteins and we can consistently generate these proteins that are of a consistent quality, okay, then we should be able to make lots of reagents that are much more consistent from batch to batch. So if we look at this timeline, the first recombinant reagents were actually started back in 2003. Cambrix and now Lonza introduced their recombinant factor C reagent in 2003. In 2011, Hyglos launched their test, EndoLYSA test. 2013, Hyglos bioMérieux launched their endozyme recombinant factor C test. In 2015, Segugaku, which is the parent company for Associates of Cape Cod, launched the PyroSmart test, and that is a full recombinant LAL test. It's not just recombinant factor C, it's full LAL. In 2018, we have Hyglos and bioMérieux launched Endozyme to Go. That's a little bit different twist on the test, but essentially the same factor C test. In 2019, Associates of Cape Cod reports on the recombinant LAL reagent that they have at the USP workshop that happened in June, and at the same time, Charles River Laboratories ended up launching theirs as well. They described their recombinant product at that same meeting. So, this has been a while in the making. It started out in 2003. So when you say, well, why didn't it started out in 2003? Why didn't it catch on? Well, the problem back then is that when it started in 2003, the Lonza, then Cambrix, went to FDA and said, okay, where are we going to have this regulated? And in the end, FDA said, well, we're not going to regulate it. It's not a blood product. It's not a medical device. It's not a biologic. It's not really anything. So there is no regulation for it. And so what happened was it was called an alternative test, because at that point, all of the reagents used from the horseshoe crab blood were qualified licensed reagents. The recombinant reagents are not licensed per FDA, and that makes a big difference in terms of, especially back in 2003, in terms of the marketing. Okay, in terms of the marketing of these reagents, people were reluctant to use a reagent that had not been approved prior by the Food and Drug Administration. So a little bit about the bacterial endotoxins test. Our Chapter 85 is harmonized with the Japanese and European Pharmacopeia. Right now, in all three of them, the use of the recombinant reagents is considered by the Compendial Chapter to be an alternate test to the BET. Now, what does that mean? When you look to validate an alternate test, you have to go back to the USP and you have to look at two chapters, 1223 and Chapter 1225. 1225 is a validation of alternate tests, but it's more chemically oriented. 1223 is the validation of alternate microbiological tests. And basically, validation of the test is a comparability. It says, does this test give me the same or better answers as the compendial test? Same or better, meaning within an error, because we know we're not going to get exactly the same answers. We're within an error. Are they going to give us the same answers? Is it going to be faster? Is it going to be more cost effective? Is it going to be this, that, and the other thing? So we have to run a comparison. So that validation really is a comparison of the new method to the existing one. FDA, in their 2012 question and answer, also said that it was an alternate test. Now, we know that FDA has proved at least one product for testing with recombinant factor C, and that company submitted a whole load of data basically discussing the comparability of the recombinant factor C with the existing LAL test. That's not trivial. The European Pharmacopoeia issued a separate chapter 2.6.32 back in January of this year. USP issued a proposed revision to 85 for comment in September of this year, and the Japanese Pharmacopoeia issued a chapter that would keep it an alternate test back in October of 2019. Now, if you notice, the European and the Japanese Pharmacopoeia are both considering the test at this point to be an alternate test. The thinking here, the way we're thinking at USP is if the test is in fact comparable, then why should it be alternate? So if in fact the test is deemed to be comparable, then why not include it in the regular standard chapter? So what we've done is we've assumed for right now that the recombinant reagents will provide us with data that will demonstrate the equivalency of the recombinant test with the compendial BET. And you know, again, as we look back at it, we would think that the test methodology really is not different. The recombinant factor C is a fluorometric test, so the reagent is a little bit different. It's got a different formulation, and you need a fluorometer to read the test. But in fact, it's really no different in how you run it than is the endpoint chromogenic test. And if you look at the recombinant LAL, it's really no different than the current Chapter 85 test. And so we're kind of looking at it to say, okay, we're going to assume for the time being until we know otherwise or learn otherwise that these tests are all going to be equivalent. So what is the thing here? So if the recombinant reagents are shown to be equivalent for the detection and quantification of endotoxins, then we think they should be included in 85. If the recombinant reagents are not shown to be equivalent, all right, then we think that they would require product-specific validation. In other words, they would maintain themselves as alternate tests. So what do we need to do here? So we have to say, if the BET chapter is revised, one of the challenges is that it will contain both regulated and non-regulated reagents. And so from the user standpoint, what does that mean? So that means from the user standpoint that the vendor management of the non-regulated reagents becomes basically 100% the purview of the user, because FDA is not going to inspect those facilities for GMP or any other kind of compliance. So then the non-regulated reagent, wherever that comes from, would then fall on the user. The users will still have to show suitability with the product. All right, that suitability is otherwise known as interference or inhibition enhancement, as people used to call it. Okay, we still need to do that. And we understand that the interference patterns might be different with the recombinant reagents than they are with the standard LAL reagents. How do we know that? Well, we know that even with the current standard LAL reagents. If you do a suitability test with one reagent, you might find out that the non-interfering concentration is different than if you used it with reagent B. So because of the formulations even of the existing regulated reagents, we know that there could be different interference patterns. And so we would expect certainly that there would continue to be different interference patterns here. So that's the things we would need to take into account. The USP proposal is out for comment. I believe that the comments closed at the end of October, but there is still time if you want to make a comment to the USP proposal. Please feel free to do so. One of the things, though, this is the sort of caveat, is that USP really needs to see data right, in order to understand comparability. And so in order to make the proposal official, in order to change Chapter 85, we have to have data. So what do those data have to be? Right now, if you look at the published literature, the data out there are quite good, but they're missing one thing. The data out there generally right now compare things with no endotoxins activity. So you test your product and there's no endotoxins activity in there. You test it with recombinant factor C or recombinant reagents, and then you test it with LAL reagents and you get none detected in both. You test your water for injection with recombinant reagents and you test them with regular LAL reagents, you get no reaction on both. And those are the kinds of data that are essentially out there right now. So what really do we need to understand in order to understand comparability? Well, we need to understand that if a product is contaminated, would the recombinant reagents in any of their various forms give us the same answer as the LAL reagent? The LAL reagent is the benchmark. It's the gold standard because it's the approved compendial test. So we can't always compare 0 to 0 to 0 to 0. What we need to understand is if we get 10 EUs with some naturally contaminated product, 10 EUs with the LAL test, do we get within a margin of error 10 EUs on the recombinant test? Now let's think about this. If in fact there's no endotoxins activity in here, it probably doesn't make a difference what reagents you use. But if there is endotoxins activity, and if there is a difference in the detection and quantification of the activity depending on the reagents that you use, especially as you approach the endotoxin limit, there is a big difference can happen. You can either reject product that's really good, or you can accept product that really exceeded the limit. And so what USP is looking for now is data on materials where endotoxins activity is there. We want data where endotoxins activity exists. Now, you can say, well, we can put some RSE or CSE in there and then compare it. Well, you know, there's a significant controversy going on right now about the appropriateness of a spike wound recovery with the RSE, CSE versus something that's more representative of what might be in the product. So what we would like to see is data on actual product, contaminated product. So you're going to say, well, Karen, my company doesn't make contaminated product because we're really good. And I think that that's great and to your credit, but there is contaminated stuff in your facility. Try the upstream water system. The pre-treatment water system, generally after the carbon filters, certainly the upstream pre-treatment part, very often has places where there are endotoxins that come into the system and where the bacteria set up in the carbon filters, for example, they might set up housekeeping and produce endotoxins. Comparator testing for LAL reagents should include a glucan blocker. Now, why is this? Well, if you remember back when I showed you those cascades, the LAL reagents are responsive to glucans. Glucans can come in and they can actually sort of downstream mimic the activity of endotoxins, right? The recombinant reagents are glucan resistant. There is no possibility of the glucan pathway in there. So if we're going to compare the LAL reagents to the recombinant reagents, then we have to make sure that we block the glucan pathway in the LAL reagents. Here's the announcement that we put in USP about the change. You can see here that the USP Bacterial Endotoxins test, we put in here change to read, okay, and we changed all kinds of stuff in there. And it's really not much. We just changed basically, you know, to include the use of a fluorimeter. We included the use of, we took out the use of anything that says LAL, and we called it now recombinant, either LAL or recombinant reagents. So what we did is modify the text, which really didn't take much modification in order to accommodate the use of these tests. Now here's the one kicker here, where it says, notice, documents in PF Online are not official and they may never become official. So in our case here, where we're looking for data to demonstrate comparability, this particular chapter may never become official if, in fact, we don't see that comparability, okay? And or if it may take a while in order to accumulate enough data in order to make a statistically valid conclusion from the data that we do receive. So we would ask that interested parties comment on the proposal and provide comparability data, and preferably the raw data. And we would like to see everything, and we would like to see stuff that you chose to invalidate, and we would like to see stuff that had valid system suitability around it, because we really would like to understand what's going on with these reagents, if in fact we can confirm that they are comparable. With that, I think I'm going to turn it over to Veronica.
Thank you, Karen. This is Veronica Wells. Good day to you all. Thank you, Karen, again for such an informative presentation. In my portion of this webinar, I will be focusing on demonstrating practical examples of the use of the recombinant reagents, and more specifically, the recombinant LAL Cascade, and including an example of some equivalency testing. Just to start off with a quick disclaimer as well, this presentation is shown here for educational purposes only, and any decisions and actions should be based on relevant regulations, not on this presentation. Karen mentioned in her part that Seika Gakko, who's the parent company of Associates of Cape Cod, launched the first recombinant chromogenic reagent for the use in the BET testing back in 2015. The name of this product is PyroSmart, and it is commercially available on the Japanese market. It's been also included in multiple collaborative studies and presented on at PharmaLab in 2016. So I will talk about this reagent today. This is what that reagent looks like. It comes kitted with a reconstitution buffer. And in order to use this reagent as a kinetic chromogenic test, using a regular absorbance plate reader, the data, absorbance values at 4 or 5 nanometers would be collected over a certain incubation time at specific intervals. The data would then be reduced in 2 different ways, either as an onset time assay, which is globally used, and that's the one that Karen was referring to earlier, or as a mean rate assay, so v-mean. Either one of them have been used and have been actually validated. So this presentation is going to go through the basic features of this PyroSmart reagent, qualification, as well as some proficiency testing, screening for interference, and side-by-side comparison of materials that were actually contaminated with natural endotoxin. And towards the end, we will touch on some publication review. So the basic feature is actually very simple. The reagent comes with a volume of buffer, and basically a volume of reagent that is sufficient to perform 50 reactions, so 50 wells on a microplate, standard 96 well microplate. So 50 wells are to be used out of a vial. So in order to run a full microplate, we would actually need to use 2 vials. Each well will take 50 microliters of the reagent, and that gets combined with 50 microliters of the test sample, whether that's water for negative control standards or samples. The test prep is very easy, and it's extremely familiar to those who have been already doing kinetic testing, whether it's chromogenic or turbidimetric. And the lowest limit of quantification goes down to 0.01 EU per milliliter, and it's achievable within 30 minutes if we choose to use the v-mean assay. So this kind of links it back to, is it equivalent? Is it the same as the conventional LAL assays, and here it would be equivalent because the LAL assays also achieve 0.01, but it's much longer time. A huge advantage of this reagent, as much as any other recombinant reagent, is there is no cross-reactivity with the beta-glucans due to the missing factor G. So quick lab setup here. So as usual, we would have to prepare a series of standards, series of samples, then transfer them onto the plate, then reconstitute the PyroSmart reagent. And as soon as the microplate reader is up at 37 degrees C, the reagent would be added to the plate, and then a 30-minute assay would be started. And at the end of that, software, whatever you use, would then be used to actually interpret the data. So for either type of assay, whether it's onset time or mean, 30 minutes is the runtime. And so combined with the prep time, which obviously could vary depending on how many samples we do, the test result is considerably shorter. I am usually able to get my results in within 45 minutes. So that would be considered to be a considerable high improvement. So this here shows a standard curve plot that would be performed as part of the regular initial qualification of the reagent, most likely on multiple vials and either for multiple lots as well. So it shows actually the data reduction here as an onset time. You can see the correlation coefficient perfect with the minimal value being 0.980 as per the Pharmacopeias. And so this actually represents the standard curve, wide range curve from 50 down to 0.05 EU per milliliter. On this slide, and this refers back to, again, what Karen was also mentioning, is the lot-to-lot consistency of the recombinant reagents. So generally, it is expected that between testing different lots and testing them over a certain amount of time, they should be more consistent than the classical LAL reagents. And the reason for that being that there is no more use of the blood of the horseshoe crabs here in the production process. So you can see that, especially on the lower end of the standard curve, the variability usually does happen, where the onset time could vary between lots by hundreds of seconds at a time. You can see here that for 4 different lots tested over 6 months, the variability was actually very low, and then the onset times for the 0.05, including 0.05, were actually very, very close to each other. And the slope and R value were reproducible as well. And this here shows how the PyroSmart assay compares in terms of the onset time against the kinetic chromogenic and kinetic turbidimetric assay. All of these tests were performed with exactly the same standard series preparation, and it was the same range of curves of standards. And it's obvious that the PyroSmart is much faster. Again, the results are achievable within 30 minutes, and that does include a separation of negative controls versus the chromogenic and turbidimetric test. And because those could react within 50 minutes or longer, and that may change sometimes significantly with the lot numbers. So in order to actually show the comparability of the recombinant reagent against the conventional reagents, we use 2 proficiency samples. These are used as part of an internal proficiency testing scheme, and both of them are actually prepared with the lipopolysaccharide, so it's a control standard endotoxin, but at least prepared in 2 different matrices, saline and water. You can see the acceptable range, so basically what we were expected to measure as far as endotoxin concentration, and then a comparison of those results provided by the LAL methods versus the recombinant methods. So the PyroSmart values fall right in the middle of the acceptable ranges, and they actually compare very, very well, especially with the kinetic turbidimetric test. So that would meet the expectations of what those results should be. Also, as Karen mentioned, suitability testing, so screening for interference. Just because using recombinant reagent, it doesn't mean that interference from matrices is going to disappear for us. The interference is primarily triggered by the impact of the sample on the LAL enzymes or any enzymes in general, as well as on for example, aggregation state of the control standard endotoxin. So in this little study, we chose 3 different substances, which actually happened to be in a proposal study by the EDQM. And we ran a screening for interference on them side by side for PyroSmart as well as for kinetic chromogenic assay, which was endotoxin specific. That's what ES stands for, which means that the beta glucan response was actually blocked out there. So aciclovir as a antiviral drug, gentamicin as an antibiotic, and PVS buffer 1X as a generic buffer were used, and they were tested in 3 different dilutions. You can see that there was no interference for aciclovir and PVS for either reagent, but there was, as expected, interference for the gentamicin. And in both situations, the non-interfering concentration that was found for gentamicin was the 0.01 milligram per milliliter. So the same for PyroSmart as well as for the chromogenic assay. And that's most likely because of the impact on the aggregation state of the endotoxin here as an antibiotic. Then the next question was, how much endotoxin can we measure in these chosen drugs at the non-interfering concentration? And unfortunately, in our case, there was no endotoxin measured, so less than 0.05 in all of the situations, and you can actually see again the PPC recovery. So to sort of tap into what Karen spoke about, we decided to challenge these concentrations, these non-interfering concentrations with tap water, because tap water does contain natural endotoxin. So at the bottom, we can see the endotoxin concentration values reported for 1 to 10 dilution of the tap water. It was basically 1.8. By the kinetic endotoxin-specific kinetic chromogenic assay versus PyroSmart 1.3. So very comparable right there. And that water went into all of these preparations of these matrices. And the endotoxin was again recoverable in all of them, including the PyroSmart, exactly where it should be. So that worked very well as well. So the next part that I want to focus on here is the specificity of the recombinant reagent to the endotoxin, because as we all know, 1,3-beta-glucans do present a challenge to testing by the LAL test, and because of the reactivity towards factor G. And it may not be as known that they also present a huge source of variability in the LAL testing. They don't exactly react the same every single time, which is demonstrated on this slide. 2 different operators running 2 exactly the same tests, testing Curdlan, which is a short 1,3-beta-glucan, at 3 different concentrations. And you can see that it was tested by PyroSmart, but it was also tested by kinetic turbidimetric assay, which was actually not endotoxin specific, meaning that it will measure glucan as well. And what was not expected here is the differences that for operator 1, the PPC recovery was well over 200%. Again, that would be expected. But for operator 2, the PPC recovery was actually within valid range, especially for the 2 higher concentrations. But the result was highly variable. So this means that the variability introduced by the beta-glucan is definitely there and may not be exactly replicable between various tests. And but PyroSmart, on the other hand, produced constant results of the same results for both operators repeatedly. Also, when we tested a glucose sample that was known to be contaminated with large endotoxin and glucan, And glucan, because this glucose sample actually went through a filtration process where the glucan was introduced. This slide shows the differences in the results by a kinetic chromogenic assay, endotoxin specific one, where the result was produced as 1900 EU per milliliter, while PyroSmart's result was 360 EU per milliliter. It is believed that in this situation, the large background of glucan that was actually in the sample was not completely diminished by the use of the endotoxin-specific buffer in this case. So again, having that variability by the glucans, it's desirable to eliminate that and by using a reagent that actually does not react with them at all. Furthermore, we also tested samples that contain natural endotoxin. These were medical device extracts that actually represent real medical devices, as well as tap water samples. And again, side by side with endotoxin-specific chromogenic assay, the results are very much comparable, sometimes on the cusp of the 50%, but that could be potentially also, again, contributed to the glucan background in there, especially in the water samples. The data is definitely there, and the data will start to coming as we use this reagent more and more. There were publications, papers published on this, so 2 large collaborative studies that were done in Japan by Dr. Kikuchi's group. They both looked at a comparison of all of the recombinant factor reagents along with the recombinant cascade and compared it with 3 different kinetic chromogenic assays. So this was published back in 2017 and 18, and just recently a new publication also coming out of Japan on the assessment of PyroSmart's analytical characteristics. It is available for download, and it is very interesting because it presents a study of the interference pattern in 118 various product matrices. So it shows the interference profile in all of these products. And more on this study is going to be presented at PharmaLab next week in Germany. So in conclusion, I'd like to mention that this obviously recombinant chromogenic reagent may be one of the options to be used as an alternative test to the classic LAL reagent, and that's thanks to its ease of use and familiar process, familiar setup. And in order to generate the data that we can then provide back to the USP, to show the equivalency with the LAL reagent, we will be more than happy to work with anyone who is interested in this by setting up evaluation studies and working with any interested parties on looking into whether this is something that they would like to pursue. Thank you for your attention, and I will be switching over to Brendan.
The first question we have says, regarding the USP wanting to see data, is there a specific study designed to follow?
That is a good question. No, there isn't. What we would like is that any design that you put together includes endotoxin specific LAL reagents, so that there is sort of an apples to apples comparison there. And again, we would like to look at things that are naturally contaminated. So, you know, any products that you might have or any materials that you might have that are naturally contaminated, we would like to look at those. And we're going to have a statistician take a look at all of these data. And in fact, USP is going to put together a smaller study within ourselves and look at this as well. But there's no standard study design.
To whom should the end users submit the raw data regarding recombinant reagents?
Okay, those should be submitted to Dr. Trimuli, and his email address is RST, R like rabbit, S like Stephen, T like Thomas, @usp.org. And so if you have data that you would like USP to consider, then he's the person who would be the recipient of those data.
Perfect. And what is the timeline with the PF proposal? When should we expect the final text to become official?
So the final text won't become official until we have data that basically support the proposal here, which is to include all of the recombinant data in Chapter 85. So it could take, I don't know, it could take a year, but the sooner we have data, the better we are. So it could take some time. And, if we can get data from different sources, that's also helpful because we'll see if there's something, the source-specific variability or a laboratory-specific variability that we can try and understand a little bit. People on the phone, I don't know how old anybody is, but certainly I was around back in the day when these sorts of things were being done prior to inclusion in the USP. Back at the very beginning of the LAL time. So, what we're asking for at this point really is not considerably different than the kinds of data that were generated back in the day.
Is the ACC product RLAL or RTAL?
So I will take that question. So the PyroSmart product is RTAL, so Tachypleus. And that's because it was developed in Japan where the Tachypleus reagents are being used.
Great. And will the origin of the recombinant factors, the species of horseshoe crab, no longer be of interest, or can all species of horseshoe crab be used, also from a regulatory perspective?
Yeah, I can't speak from a regulatory perspective because I'm not a regulator. Certainly the reagents that are out there now are from 3 different horseshoe crab species, which is another thing that we need to take into account. So, we know for a fact which horseshoe crab species, which of those proteins are now in these recombinant reagents. And so if we see, for example, that, the limulus gives us a different, reaction than Tachypleus or Carcinoscorpia, we can look to see if there's a difference in the source as well.
Faraz from Pakistan says, are there recombinant reagents for gel clots too?
As far as I know, they are not, and that would be difficult to manufacture due to the very low signal that actually the gel clot reagent produces. It's a much lower signal. So far as I know, the focus is on manufacturing kinetic reagents only.
The difference in results between traditional LAL with ES buffer and the PyroSmart, how is that not a larger concern? And which result is correct?
There was a large difference in the glucose sample, which we knew by testing the glucose sample, but a glucan-specific reagent. We knew that the glucan background was extremely high in there, and that glucan background can then may not be blocked completely by the use of the endotoxin-specific buffer. So that was the thought, and that was why I actually included that in this presentation as well, to present that in some extreme cases where the glucan background is high and it's always variable in terms of the molecular weight and branching, the glucans do react differently, and they may not be completely blocked by the use of the endotoxin-specific buffers. But on the other hand, with PyroSmart, since there is absolutely no reactivity at all, the lower signal is more likely to represent endotoxin only and not endotoxin and partially glucan as well. But of course, this study that I showed here certainly does not represent any statistical data. These are just different scenarios that are trying to explain the differences, the variability in the testing that the glucans do cause.
To what degree does product-specific suitability need to be established in order to use an alternative method using recombinant reagents? Replicates? Question mark?
With any LAL test or with any bacterial endotoxins test, the product suitability part, meaning the inhibition enhancement part, has to be demonstrated. So even with the test that's currently in the compendium, so remember, those tests that are currently in the compendium are considered to be de facto validated tests. And so what you have to do, what we have to do as users, we don't have to revalidate the test. We have to demonstrate that the test, the valid test, is suitable for testing our product. Now we know, okay, from history, or at least certainly I know, I'm old enough to know, that if you take the same product and you test it on different lysate manufacturers' reagents, you're likely to get different interference patterns. And that most likely is due to the fact, Veronica, please help me if I'm wrong here, is that while all of the lysate formulations do the same thing, they detect endotoxins, the formulation components of the reagents are proprietary. And so different reagents from the different manufacturers actually have different formulations. And so that's why we get different interference patterns. You will not lose the requirement to have to run a suitability test with the recombinant reagents.
She was specifically talking about release testing.
Well, you need to do suitability on release tests, but you also need to do it on your in-process samples. Arguably, that's just as important, right? Because you need to make sure that the particular components and the particular formulation of in-process samples may be different than the finished product samples. And so if you're using the bacterial endotoxins test as a surveillance tool throughout the manufacturing, you need to do that suitability testing on each of those as well, because they're essentially different formulations. So I think it's kind of the same thing.
Would naturally contaminated samples be clean samples reformulated in tap water or contaminated by NOE from various organisms?
There's a lot of ways, I guess, to make contaminated product. Preferably, we would like to see real contaminated product from real issues going on in the real manufacturing. The good news here is that the pharmaceutical industry is under very good control, and we don't normally have those problems. So sort of simulating those problems by using tap water or upstream water from your pre-treatment water certainly would provide some level of natural contaminations in there. NOEs are made in the laboratory, and so NREs, even though they're called naturally occurring endotoxins, they're really not natural in the sense that they are still prepared in the laboratory. So using water that is naturally contaminated certainly is a step closer to doing this, and using tap water to either formulate the product or perhaps in the end reconstitute the product might be appropriate. That might help. You remember organisms adapt to their environment. So if you were to find an organism in your water for injection system, it's highly adapted to that particular system. Or if you find a gram-negative organism in your product, it's highly adapted to your product. And so, we can't anticipate all of those things. So we have to think about what might be a reasonable alternative. And I think using either tap water, but you have to remember there's chlorine in the tap water, which could also cause a problem. Okay, so if you use the upstream pretreatment water post-dechlorinization, then I think that would be closer.
PyroSmart will be used in 1:4 dilution or 1:1?
So it will be used, or it is currently used in the 1:1 ratio, 50 microliters of the reagent plus 50 microliters of test sample.
When requesting data, is the USP also asking for identification of the organisms from naturally contaminated waters?
Certainly that would be helpful. That certainly is not required, but it's certainly helpful. There are some gram-negative bacteria that don't have endotoxins, and so it would be interesting to look and see if you have a naturally contaminated product. The endotoxins have to come from some sort of bacterium at some point in the product stream. So if you can identify where they came from, then that would be helpful, but I don't think that at this point it's essential.
Gel clot will remain the final test in case of dispute as per USP, can't really read the numbers there, even after recombinant.
Yeah, it did. If you read the proposed chapter in the Pharmacopeial Forum there, if you read that proposed chapter, the gel clot, really nothing changed in there. The only thing that changed in there is really genericizing the instrumentation and genericizing the lexicon for the reagents to include the recombinant ones. But the reference to going back to gel clot for the ultimate reference is still in there. Now remember, gel clot, and this is one thing that people get confused. Can I just, Brendan, the gel clot test, when it's in there as the referee test, doesn't mean that if you have a kinetic test, or in this case, a recombinant test, that fails, that you can go back to gel clot and see if it passes, right? That's not what that is meant to do. It's meant to say if you get a different answer than a regulatory agency, let's say, who tests your product, and we can't resolve those differences, then we go back to gel clot. But it's not meant to either invalidate or validate a test that you've produced in your laboratory.
Is there inhibitor activator for RFC method?
Well, that kind of goes back to the whole suitability testing as well, that basically the recombinant reagents are still enzymes. And so as enzymes, they have to be used under optimal conditions. So any suboptimal conditions, pH, ionic force of the solution, the presence of detergents, will certainly still inhibit them. There certainly may also still be a situation, for example, in the presence of surfactants, there may be enhancement that happens. So that would be, I guess, the activator in this question. But that, again, goes back to the fact that the suitability testing, continued suitability still has to be performed as it is with the classical LAL reagents.
Well, perfect. That's all the time we have today. We'd like to thank Veronica and Karen for sharing their knowledge with us, and also offer a special thank you to Associates of Cape Cod for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleagues. So thank you, Veronica and Karen, for joining us today, and everyone else out there, thank you for joining us, and have a great rest of your day.
Thank you, everybody.